Welcome, everyone. We're going to get started in about a minute. We're just waiting for folks to be able to be connected into the webinar room. Okay, I'm going to get started, Jen. Okay. Welcome to our second session on discussing gene therapy. This session is titled Lessons Learned from Gene Therapy Clinical Trials. In the previous session, we reviewed the basics of gene therapy and how programs are designed and developed. If you missed that first session, you can view the recording through the Symposium app. Thank you for the questions already submitted. We have incorporated them into our Q&A. Jen will be monitoring questions submitted during the presentation, and we will include as many as possible. At the end of this session, an email will be provided where you can continue to send in your questions. Our goal in hosting this session is to provide an opportunity to hear from experienced researchers and clinicians in the industry. Tonight, our panel of speakers include Grace Pavlov, Vice President of Research at Avanti Bio, Meredith Schultz, Executive Medical Director and Head of Translational Medicine, and Matt Klein, Chief Development Officer at PTC. Emeritus at Novartis Gene Therapies. And I'm joined by Jen, the CEO of FARA. Just to start off, I want to say to the panelists that we are so grateful for your time and expertise in educating our FA community tonight. So welcome to you.
Thank you.
Just wanted to say and review the aim of our discussion today is to cover high-level principles regarding development of gene therapy programs and clinical trials. During this discussion, the panelists will not discuss timelines of when gene therapy clinical trials are expected to be available for FA, and they won't be discussing specific details about any program that's specifically in development, including expectations for eligibility criteria and data collection as it relates to FA. So I just want to start off the first. Let's discuss a little bit about how gene therapy clinical trials are different from what the FA community knows as small molecule drug trials. So kind of the tradition of what we think of for a clinical trial versus what we might expect with a gene therapy trial. Grace, can I ask you to go through some of these features that are on this slide and give us some examples, maybe from your own experience?
Sure. So in small molecules, a lot of times you will see the distinct three phases where in the initial, you're looking at safety, and then in the second, initial efficacy, and then, further larger scale in phase 3. In gene therapy trials, it tends to be more group. So in the phase 1, you can see both safety and dose. And then the phase 2 and 3 will be combined for efficacy. Again, there's no hard and fast for everything, but that tends to be the situation.
If I can just interrupt for a minute, if we think about the phases, I know that a lot of the discussion in the first session and the questions that came in was kind of that suboptimal dose idea. And so I think when people think about the phase 1 and understand safety and tolerability, but what if they get a suboptimal dose and can't get to the efficacy part?
Right.
How do we think about that?
Mm-hmm. Well, I think first off, there are a lot of studies that preclinically that go into dose selection, and you try to, it's not like it's a hit or miss and you just go in with whatever dose you want. So initially there is a lot of work to try to get in that correct ballpark. So, that would be one part. And then as you said, what if it wasn't an efficacious dose? I think people are trying different ways of trying to mitigate the immune response, even with the first trial of the gene therapy, so that people could do it again. Or there's the possibility of using different viruses in a subsequent clinical trial. I think those are all on the table right now. Like any virus that you have, there can be an immune response to it. And in fact, about 50% of adults already, because it is AAV is a natural virus, they already have some immunity to it. So as I said, there's different ways to try to mitigate that immune response to the virus. So, definitely something sponsors are thinking of and looking to mitigate.
Thank you.
Yeah.
Grace, I think one of the things Ron Bartek has been kind of saying over and over again is that, with phase 1 gene therapy, that first dose where we're looking for something that's minimally effective, right? Versus in small molecule drug trials in a phase 1, you might start below that. You might start at a very, very conservative low dose that, is your guaranteed safety kind of thing, and then slowly ratchet up. Whereas in gene therapy, that first dose, while we're looking at safety and dose, we're trying to aim for a first dose that's that we think is going to be at least minimally efficacious, right?
Right, correct. As I said, there's a lot of work before you even get to dosing in humans, working towards that minimal effective dose. Yeah.
And then I think if you can talk about the duration of trial, that's going to be a very different approach than what we're used to in small molecules.
True. So in a small molecule, you would be dosing either every day or every month or whatever that characteristic would be. With the gene therapy, because you are getting it's that one dose, there's a lot of you want to look at long-term follow-up and the durability, how long it would last for. So that's why you would have that kind of clinical trial would be a much longer compared to something that gets dosed every day or week or whatever.
Okay. And then again, same thing with the number of participants. If we look at we've gotten to it, if we think about getting to a phase 3 efficacious study, in each phase, we continue to build up that number of participants in a traditional trial. And how is that different then?
Well, the numbers that are listed there for a small molecule, again, with the rare diseases, generally you just have less people with it. So, that number is a pretty high number. And so that might be something for a more common disease, but you would tend to see less for rarer diseases. And so that would be the same case with the gene therapy trial. Again, it would tend to vary by disease, but be fewer numbers. Now, the number is going to depend on what kind of measures or how quickly the disease progresses, who you're studying in the trial, what kind of outcome measures you're using to see how efficacious the therapy is and how variable those are. And I know you're going to be talking about that in a little bit, but the number of participants really is tied into all those parameters too.
And does the number of participants have to account for variability in presentation of symptoms or age of onset? Does that?
Well, you're certainly, when you're doing a clinical trial for any sort of disease, you want to get people as homogeneous as possible for whatever you're measuring. So, because again, you want to be able to see the effects. But again, a lot of it is going to be, there's a lot of statistics works that go into this, deciding ahead of the trial, depending on what measures you're going to use to determine if the drug is working or not, how variable those are, and predict ahead of time how many patients you would need. So a lot of it is based on the variability of the measures that you're going to be using in the clinical trial.
So the number of participants, based on your experience, it's not something that is preset. It can be very disease specific.
Oh yes, for sure.
Okay.
Yeah.
Okay.
Yeah.
And then the biggest question that people always ask is about placebo. Now, this might be a little bit different in gene therapy, of wanting to get to the point of knowing that it's the right dose. But people often ask or think about, okay, what if I'm in the placebo arm? what does that look like for me? And are there options when the trial moves on to a different phase or wraps up altogether?
I know that you have yes there. I think there's different ways of doing that, and it really depends on the particular disease and the trial. One could be looking at the natural history data. There could be a placebo-controlled arm, or you could have like a delayed start so that individual be followed for a certain amount of time and see how without the drug and see how the parameters go. And then, maybe 6 months or whatever time they would then start the therapy. So they're kind of their own control in that. So there are just different types so it's hard to generalize here just based on the type of whether it's a small molecule or a gene therapy. I think it would be really dependent on the specific study and what the parameter is there. But a lot of times,
that is different.
Yeah.
Okay.
Yeah.
And then again, too, with the number of study sites, I would think that smaller number of patients means fewer number of study sites, but
could, and remember that it's going to be more that in order to deliver the gene therapy, there's probably going to be a more limited number of sites that are trained and have all the facilities to be able to do that. So there might be smaller number of sites than for something that's, dosed orally or injected in a different way.
That makes a lot of sense. Good. Anybody want to add anything from their own experience? Okay.
Yeah, and I was just going to say, part of the reason too, for there being less patients in general in gene therapy trials is one, because gene therapy right now is really not very easy to manufacture. So it's hard to make. The other aspect of that is that we are also hoping that gene therapy will have a big effect size. And so when you have a therapy that has a large effect size, you can, sometimes not have to use that many patients. So just wanting to add a little bit of detail there.
Yeah,
excellent point.
That's a good perspective. Yeah.
Yeah, I'd also add that I think all of these considerations, in particular placebo control, the number of study sites, is also going to be impacted on how the gene therapy is administered.
Right.
Gene therapy can be administered intravenously. It can be injected like a spinal tap into the spine, or it can be administered through, say, a very intricate surgical procedure. And so obviously, is based on what the mode of administration is, you may have fewer centers that actually have the expertise to do, say, a specialized surgical procedure. That can influence the number of study sites. Also, there are big questions about, a placebo when there's a surgical procedure. I mean, we're used to oral drugs where giving a placebo or giving, what people say is a sugar pill, something that looks a lot like the drug, but it's just a pill. Well, that's a lot less invasive than undergoing a surgical procedure and not receiving the gene therapy. Now, there's a trial now for Parkinson's disease, for example, where there's a treatment group for gene therapy. It's an injection to the brain, and there actually is what's called not really a placebo group, but another group that just gets a surgical procedure, part of the procedure without the gene therapy, which seems pretty invasive, and it is. But again, all of this goes towards the need for us, the requirement that we have to demonstrate that a therapy is truly effective and that changes are related to the therapy itself.
Yeah, it sounds like that would, based on when we do get to gene therapy trials for FA, that there would be a lot of education around that. Once that mode of administration is developed, what does that mean then for that participation?
Yeah.
So you've given us some good things to think about. I want to move on to talking a little bit more about the specifics of the patient experience in a trial. And if I can call on you, Matt, to talk us through some of these parameters in this table and comparing. Let's start with the factors influencing inclusion, exclusion criteria, because those could be very, very different for gene therapy.
Yeah, it's a great question. Obviously, every time we do a clinical trial, there's always specific criteria that allow us to try to get to, as Grace said, really as similar a group of patients as possible. Now, while Friedreich ataxia, everyone with Friedreich ataxia has a mutation in the same gene for taxin, we know that symptoms can always be different. Age of onset can be different. And when we think about organizing a clinical trial, whether it's for a small molecule for gene therapy, we think a lot about what's the right age group to have in the trial. Is there a certain number of GA repeats we want in the trial? Age of onset, disease severity at the time of trial, all of which is done to really identify that group of folks that we think the therapy is going to work best for, and also gives us the best chance in the clinical trial to do what we need to do, which is to demonstrate that a therapy is safe and effective. So for gene therapy, we're going to have very similar considerations in terms of what's the ideal age group, what's the ideal range of disease severity, say a FARA score, for example. Are patients able to be ambulatory or not? And then there's going to be a number of other factors that are going to come into play that are unique to gene therapy. One of them is this question about you have surgical hardware placed there. Well, it depends on what kind of procedure is going to be performed to deliver the gene therapy. So for example, we've worked on gene therapy programs where an MRI is often used to help plan a surgical procedure to administer gene therapy. Well, if you're going to have an MRI, there's going to be considerations if you have certain hardware. So that may come into play. And then you mentioned viral load. So one of the things to know about gene therapy, when you think about gene therapy in general, and it's really in its simplest forms, there's three key considerations. It's the gene that you're providing, right? So in this case, we're going to be giving frataxin. The target, where you're putting the gene, are you going to inject it into a vein so it gets to the whole body, or we're going to be injecting it directly into the brain, given that there's parts of the brain that are the source of a lot of the disease in Friedreich ataxia. And the last is, how's the gene going to get there? It has a vehicle, something that actually takes it to where it needs to go. And as Grace alluded to, what's usually used as that vehicle is a virus particle. Now, I want to be careful, especially now more than ever, given COVID, to say that we're not going to be injecting people with live viruses and giving people a virus. But what we are using is components of a virus. The virus can't replicate, so we took away its ability to actually replicate and cause a viral infection, but it's still a virus particle that's really sort of the UPS truck that's delivering the gene to where we want it to go. And so that virus particle has its own characteristics that can stimulate a response from your body. Your body may recognize that virus particle as, alert virus. And if your body has seen that type of virus before, it may already have antibodies to it, already used to, it's on extra special lookout now that it's seeing it again, and that can cause an immune reaction. So based on a specific type of virus vehicle we're going to use, we're going to want to test folks to see if they already have seen it before, and are they at risk of having an immune response, which could be quite serious. So that's actually something really unique to gene therapy that we never think of when we're giving people, small molecules or capsules or tablets or liquids. But it's something that we really need to think about, because obviously there can be consequences for someone if they've seen this type of virus we'd be giving with the gene therapy.
Are there any measures that a patient can take that could decrease their chance of immunity for the viral capsids that are used in gene therapy?
Well, I don't think that there's, usually if people have been exposed to them, it's just through purely life. We're exposed to things all the time. And now there's certain things that we can think about doing. We can measure different levels of your response to that virus. And there've been clinical trials where folks are given steroids, for example, or other immune suppressants to actually limit the immune response. And then the other things that we think about is, you can choose different vehicles to drive that gene therapy, different virus vehicles, and some that may be less common and that there's a lower probability that people have seen before.
Matt, I wanted to just follow up on something you said about the immune suppression. I think sometimes people hear that and think different things, but also what you're looking at, it would be to temporarily decrease the immune response. You wouldn't, forever not have an immune response.
Yeah, great point, Chris.
I'm going to interject. I have a question from Scott Huckabee, and this is back to sort of the hardware issue. And he's asking, if you have rods in your spine from scoliosis surgery, would you be excluded from a trial that delivers gene therapy through a spinal tap or lumbar puncture?
Yeah, that's a great question. I think it's gonna depend. I think on certainly the location of the rods, where the injection is going to go, and then also obviously the issue around MRIs and imaging studies as well. If it's a gene therapy going somewhere else, those could be issues as well.
Yeah, I think it also probably depends on the type of rods people have, right? There are different types.
Yeah.
That are, Meredith, I think you were gonna.
Yeah, I was just going to add, in the, just something to think about is in the spinal muscular atrophy community, when I was practicing in academia, we got very good at being able to administer, drugs, into the intrathecal space, through a spinal tap for, patients that had all kinds of hardware and rods and that sort of thing. So, it doesn't necessarily mean that, you won't ultimately be able to get the therapy. Sometimes there are some stricter criteria for clinical trials. And so, that may be, different ultimately than a treatment, but, just because you have rods doesn't necessarily mean that you're not going to be eligible.
That's a good point. Thank you. Let's move on to the modes of delivery. And we kind of talked about that a little bit, both in the last session and in this session. But in terms of that mode of delivery, it's really based on the target tissue in which you want to get to. And so one of the questions that has come up more than once now has been, in the choice of mode of delivery, is there a chance that that delivery can result in too much frataxin that ends up being made in the cells? Is the mode of delivery kind of controlling for that in any way?
It's a good question. So when we think about mode of delivery, one of the key questions is, where do we want the gene therapy to go? Right. And so I think, in FA, there's been a lot of discussion about delivery to the brain, because obviously the brain and the cerebellum in particular is very important in Friedreich ataxia. Also the heart, because we know that cardiac disease is obviously highly morbid and quite serious for people with Friedreich ataxia. And so when you choose your method of delivery, it's usually related to where you want it to go. For example, we've been involved in studies where we're targeting the brain in certain diseases because we know that that's really sort of ground zero for the disease. And if you're going to want to provide a gene that's going to help cells, you want to go to the place where it's going to likely provide the most benefit. In terms of generating a dose and too much or too little, that's a really good question. And as Grace said, we do a lot of work before we even get to the clinic. And I think one thing that we can all say very clearly is that the FDA has set very, very strict standards and has very high expectations for what we're going to be, what information we have before we'd be allowed to ever treat a person. And so we rely on a lot of animal studies, and these are animal studies that provide really valuable information about the safety of the gene therapy that we're giving. Often it can be very important in terms of the safety of the procedure we're going to use to deliver the gene therapy. And of course, in that as well is going to be how we're going to figure out what's the dose of gene therapy to give. How much frataxin do we give? Now, there are publications and there's been a lot of discussion about what is the normal level of frataxin, how much frataxin does the cell need to have in order to be quote unquote normal. And obviously we spend a lot of time trying to make sure that we develop a gene therapy that can get and a dose that gets enough frataxin to get someone close to normal. But of course, it's going to differ from person to person. There's going to be other factors based on just not only you introduce the gene therapy, how many of those genes get to the right place, how many of them function once they're there. So it's not always an exact science. And that's why your question is very important. How do we think about not too low, not too high, or just right? I think the question of too high has been asked. I think Helen Puccio and her group has done some very nice work where they've shown that if you're anywhere in sort of the below 9, it's sort of different levels. They said if you give over 20 times the normal level of frataxin, that's probably bad for cells. If you give 9 times or less, that's probably okay. That's definitely safe. And between 9 and 20 is probably okay, but closer to 9 is better. And so we have some guidelines. But one of the things that we obviously are going to look at in all of our studies before we even get to people is this question of dose. And we're going to see what's going to happen when we're giving a lot, what happens to cells. And that's one of the things we're going to have to justify when we go to the FDA and say, okay, here's the dose for the 2 doses we want to test in people. And it's with assurances that it's not too low and it's not too high, sort of trying to reach that Goldilocks level just right.
So if we're talking about not having it be too low or too high, are those studies also giving evidence about the percentage of cells in that target tissue that need to receive a dose in order to see a therapeutic effect?
Well, typically what we do, so this gets actually to a lot of very interesting issues and challenges in Friedreich ataxia, which is, we often do these studies sometimes in, rats, pigs, non-human primates that don't have Friedreich ataxia, right? So it's not always clear that we can tell right away that we've actually fixed Friedreich ataxia. But what we're able to do in some of these studies is say we're going to give the gene therapy to a part of the brain. We're able to, during the animal studies, see how much of that gene is getting expressed. What are the levels in the cell, per cell? How much are we expressing? What was the health of the cell? So we're able to get a lot of that information that all goes into our calculation of the dose we're going to give to people. And I think it's important to mention, this is why it's, I think probably, well, I know everyone's anxious for gene therapy, right? And people, and we've been talking about gene therapy for years in Friedreich ataxia and in other diseases as well. And I think what you're hearing is that part of the reason it takes so long is there's a lot of work we have to do to make sure we get it as close to, right and safe as possible before we can come into the clinic.
I think that's what I've appreciated so far about listening to the expertise in these panels, is that when you talk about the concept of gene therapy, it looks pretty straightforward on paper, but this is definitely conveyed for us the amount of science that has to be done in each disease to make it work and to make it safe.
Yeah, people have heard me tell this story from when I was in medical school, now a very long time ago. We had cloned the human genome and everyone declared it's now going to be simple. We can just give gene therapy and cure all diseases. And, that was over 25 years ago, right? And so we're now at a point where we're seeing that the science, there's so much science that goes into achieving the promise. But I think everyone can be rest assured that we're all working night and day and as hard as we can to get this pushed forward and get safe and effective gene therapies for the FA community.
I think the other thing, and you touched on that, was that it's different diseases, but also different tissues in the body behave differently in terms of how well they take up these viral vectors, how many cells can carry the virus. So there's that further complexity also. So it's not just, if you're dealing with the heart versus a liver versus a kidney or, muscle, skeletal muscle.
Thank you. We did talk about a little bit of the placebo issue, and it sounds like we kind of have come to that conclusion that it really depends on how the mode of delivery is going to be that would determine what that placebo arm looks like.
Mm-hmm.
Yeah, and quite frankly, what the regulatory agencies are going to want us to do, right? I mean, I think that's actually a really big part of it. I think if we, we have to obviously reach an agreement with the regulatory authorities about what's the level of evidence we need, to satisfy you. And I think one of the great things about the Friedreich ataxia community is that you've done such extraordinary work on natural history. So there is a lot of natural history data that I know we're going to be, that we'll be armed with, and we'll also arm ourselves with the wonderful leadership of FARA and the KOLs to come and help us make an argument that, look, we know what this disease looks like without gene therapy, without a therapy. The other possibility is, there's been so many trials in FA now that have placebo groups. One question is, can we look at borrowing existing placebo data? So you have natural history data, existing placebo data. Can we create a good enough picture for the regulatory authorities of what FA looks like without a gene therapy, that they can be comfortable that the benefits observed in a clinical trial with gene therapy are because of gene therapy? And those are all the kinds of things I'm sure, Grace and Meredith, that we're all thinking about all the time. How can we do this using the rich existing data?
Thank you for that perspective. It's good to know that we've laid some groundwork that is extremely helpful to this planning.
And there has been a lot of discussion in other diseases and with the FDA about using, the natural history and with an eye towards just moving things faster. And so it is a very timely topic currently.
Yep.
I just wanted to say, without, sorry, Susan, without the work that all of you did, on the natural history, we wouldn't even be able to plan these trials. And so those were so important. And I know you all, contributed to that. So it's really the groundwork for, getting these, gene therapy trials to move forward. So
that's wonderful to hear. Thank you for all three of you reinforcing that. So appreciate that. Matt, when we were planning for this session, you used the word ecosystem when we were talking about what a study team looks like. And I really liked that word. And you talked about, a neurology ecosystem versus a surgical ecosystem. Can you flesh that out a little bit in terms of defining ecosystem?
Yeah, I think part of it, I think we're all pretty used to, oral, small molecule trials where, there's a company or someone ships the drug to the pharmacy, they have it in a jar, it's labeled bottle A, bottle B, you go to the clinic, you meet with the study team, they do a bunch of assessments, you sign a bunch of forms, a bunch of paperwork, some blood tests, here's your pills, go and we'll see you in a couple of months. I'm oversimplifying it, but that model of who you see and who's involved for a small molecule is pretty well known. But now when you think about a gene therapy, there's actually a lot more that could be in place. And again, a lot of this will depend on what's the mode of administration. So in trials, for example, or in gene therapies where we've delivered the gene therapy to the brain, well, now you're talking about a surgical procedure. And I know a lot of people in the FA community have had surgical procedures before. That's totally different, right? Nothing to eat past, nothing to eat or drink past midnight. You're going into a surgical procedure. There's an anesthesiologist, there's a nurse, there may be a radiologist if we're doing special images and all those things. It's just a whole nother group of people that's different from the friendly faces we may be used to when we go, for a small molecule trial. And this also takes another whole level of coordination and complexity, because you think about, you're going to have all the usual assessments. So, if you're doing a walk test, if you're having the mFARS done, if you're having blood work done, all that's still going to happen. And you may be going for at least that first time for a surgical procedure or a specialized procedure with a specialized infusion through an IV or a spinal injection. All of those things that just are a little bit different. And it makes it more complex on our side in setting up the trials. And I think for people participating in the trials, it's going to be something new and a little more complex as well.
It sounds too like that ecosystem. One of the differences is usually we go in and see the clinic coordinator for one day, or it's one or two, an overnight visit, whereas the ecosystem for, again, depending on the route of administration, could be a multiple day or much longer stay in a single inpatient kind of scenario.
Exactly. And you really have two quarterbacks, right? Because it's usually a lot of folks, you know, if you go to, you know, University of South Florida, Dr. Z is your doctor, your FA doctor, and she's going to do the assessments. And you go to CHOP and it's going to be Dr. Lynch. You go to UCI, it's going to be Dr. Pullman. Depending on where you go, that's what you're used to. But now you're also going to have the surgeon. Now, I'm a surgeon myself, so I think they're good people, but they're just different. You know, it's going to be something in addition that you're someone new, a new face for you.
Yeah.
And just another, you know, just another layer of how this is a little bit different than a typical FA trial.
Good, thank you. So let's talk now about what happens after the trial ends. What kind of options and things that we think about in terms of follow-up. Meredith, I'm going to ask you if you can work through this table with us and talk through some of these parameters.
You bet.
Yeah, so the biggest one, I think, is, you know, we're used to this concept of open label extension, and that really it goes on until the drug is going to be approved. But that's going to be probably a very different aspect with gene therapy in terms of follow-up.
Yeah, that's right, Susan. So, you know, with gene therapy, right, this is expected to be a one-time treatment. And it's expected to last a lifetime, right? And so we, you know, not only, you know, need to follow the patients, you know, during the trial, but, you know, ideally we're following patients for many, many years, you know, down the road. You know, we have data now in the spinal muscular atrophy community with gene therapy that is 5, 6 years out, and there are patients in other gene therapy communities, you know, such as in patients that have hemophilia, which is a blood disorder, you know, that's now going up to, you know, almost 10 years. And so, you know, we are, you know, are really making progress in making sure that these therapies are safe long term. But that's definitely something that we're going to be, you know, continuing to watch and watch very closely to make sure that there are not complications from, you know, the gene therapy in the short term, but also the long term. And that also goes for efficacy. You know, we are aiming to have the gene therapy be efficacious for lifetime. And so, you know, that's something that we're going to need to follow very closely to make sure that what we you know, think and what we have planned for, you know, with regards to treating the disease is continuing to work as we anticipated. So follow-up is going to be at least, you know, for you know, 5, 10, sometimes, you know, even longer than that, for years, even after the trial ends.
In the first session, we heard the word durability. And so it sounds like that's what we're defining with this long-term follow-up. Would that be correct?
Yep, that's right. So safety is one thing, and then durability. So is the gene therapy, you know, continuing to work? Is it continuing to have a lasting effect? So when we're putting a gene back in a cell, the cells, for example, in spinal muscular atrophy, you know, when we get the gene therapy into the motor neurons, those are the cells that leave the spinal cord and go to the muscles. Your motor neurons, you're really born with all the motor neurons you're going to have for a lifetime. And so, you know, with those cells, you get the gene therapy in and you expect that gene therapy to last for the lifetime of those cells. Now, there are some cells that divide, you know, they turn over, they don't have as long of a lifetime. And if you put, depending on the type of gene therapy that you get, for the gene therapy that was used in spinal muscular atrophy, which is a virus called AAV, or adeno-associated virus, that virus puts the gene therapy into the cell, But if the cell divides, then that gene therapy can get diluted over time. And so one of the things that we need to watch very closely is to make sure that the effects that we're seeing of the gene therapy are continuing to last. And, again, as Matt and Grace mentioned, there's lots of work that goes into the studies and the science beforehand to make sure that we're seeing that durability, that we're seeing that effects of gene therapy last. And, that really is our goal, is for gene therapy, to last for a lifetime. Although, it's very different dosing a mouse or a rat or a pig or a non-human primate than it is dosing a human. And so there are just some things that you don't know until you actually, dose patients. And so, we will be following, for a long time to make sure that the therapy is continuing to work. There's also lots of work going on, looking into the possibility of being able to redose gene therapy. And we touched on this a little bit earlier with, part of the challenges of that is going to be managing the body's immune response. So, being able to dose, twice with the same virus, may be a problem because your body, responds to that. But, as Matt mentioned, there may be different viruses that we can use. There may be ways, that we can manage the immune system to be able to redose. And I do think that that's going to be a possibility in the future. We're not there yet, but there's lots of people that are working on it. And so if it turns out that gene therapy lasts, 10 years, 15 years, 25 years, there's a possibility. And, I can just imagine, where we'll be 20 years from now with regards to technology and gene therapy. But, I do imagine there's going to be options in the future for redosing as well.
Wonderful.
So Meredith, thank you. Along those lines, I want to take a few questions from our audience because we have some more coming in, and I think they're nice lead-ins to maybe some of the other topics we are going to cover related to clinical trials. But one question comes up in terms of if you're in a gene therapy trial, or if you can get a gene therapy as an approved treatment, do you foresee that individual being eligible for other types of trials and or other approved therapies down the road, kind of thinking towards the future?
Yeah, no, that's a great question, Jen. And I think, you have unknown listed on the slide, and I think that unknown is a great answer, and it depends is probably another answer, right? So if you think about it, if we give gene therapy, in the setting of Friedreich's ataxia, the goal, we're really changing the disease. And, in some ways, we're almost creating a new disease. So, we're finding that out, with the spinal muscular atrophy community, right? Where you take somebody who has, really a known disease course, and we're putting, the spinal muscular atrophy gene back in. And now all of a sudden we have somebody who doesn't look like somebody who we expect to look like with spinal muscular atrophy. They're not 100% back to normal. They're much better. But now we don't really know, what their course is going to be like from then on out. And so that may affect your ability to participate in future trials, because you may not follow that normal natural history of Friedreich ataxia anymore. And so that being said, we are now to the point with spinal muscular atrophy where we are starting to study patients who have had gene therapy, and we're starting, to think about studying multiple therapies in those patients. And so we're actually looking for patients, in thinking about that community that's received gene therapy to see, is is using multiple therapies going to be beneficial for those patients? And so it may be that, we're looking for somebody who's gotten gene therapy in the past, to look at some of these other therapies. And so I think it's really going to depend on what the goal of the trial is. But, knowing that once you get gene therapy, it's quite possible that your disease might never be the same again.
Mm-hmm.
That's the goal anyway.
Yeah. One thing I think we've also always talked about is having to think about needing like a cocktail approach to therapy. And I think this is an example where that, again, SMA can be a good example, as I think, Duchenne muscular dystrophy can be a good example where there are now multiple treatments approved. In SMA, we have a gene therapy that's approved. There's an oligonucleotide, and I believe there might even be some small molecules. I could be wrong about that, but I know there are small molecules even in developments. And so, that's the way we've kind of thought about it too for FA, where we'll likely need a combination of different types of treatment approaches because we're going to have people at different stages of disease, different symptom severities, and each therapy is going to have its own, range of potential benefit. And so we've always kind of thought of it as this cocktail. And I learned a lot from looking at some of these other diseases that have come before us to try and start to think about how that might come together.
Yeah, just a couple points on that. I think that's absolutely true. I think in the SMA case, now that there is an oral therapy that people who've gotten gene therapy are receiving, and there's some studies around that certainly opens up that possibility. But I think in FA, we have a couple things that are really important to think about. One is the fact that you have different organ systems involved in the disease, right? You have the brain, you have the heart, for some folks, pancreas, and there's different regions of the brain. And so if you think about there being one gene therapy that's going to target every single organ system in the body, every single cell in the body affected by FA, it's not. It's likely not to be. And so by the different organ systems and different parts of the body involved, that alone opens you up to the need for multiple therapies. And the other is this idea of therapies working together. And if you think about what happens in Friedreich ataxia and just the disease itself, you've got cells that are really angry and inflamed and stressed from the disease. And you're thinking about introducing a gene therapy into that. It would make a lot of sense that if you had another therapy that can actually settle the cells down, correct them a little bit, they're going to be much more receptive to a gene therapy. So I think that we should also think about not only complement in terms of organ systems, but complementing each other in the same organ system where there may be a different therapy that can help a gene therapy work even better. So I'm definitely in agreement that this will be a cocktail approach.
Yeah, me too. And I just wanted to touch on as well, this gets a little bit at, the row on here that you have of outcome expected, right? So, one of the things that I think people think about immediately when considering gene therapy is, I'm going to get gene therapy and I'm going to be cured, right? It's going to fix all my problems. And I think, it's really important to remember that, that we're hoping to correct, the genetic defect in cells and tissues. But for cells, particularly in the nervous system, the brain and the spinal cord, Friedreich ataxia is a degenerative disease. And so for neurons and some of the cells that have already died, we are not going to be able to fix those cells. And so what we're hoping to do with gene therapy in Friedreich ataxia is to stop the disease from progressing. It's not known actually how much improvement we might get in the disease. And so, it is quite possible that we may have a gene therapy that helps to stop disease progression, but that we also may have other therapies that continue to help the heart to work better, or get at some mitochondrial functioning, and so I do think that even with gene therapy, it's likely that we will need a multi-pronged cocktail type approach.
Thanks.
I think there
Thank
also
you.
might be surprises, like you may deliver the gene to certain tissues, and then there may be parts of the disease that we didn't know about as much, and so we may uncover functions in other tissues then that we will have to target with other drugs also.
Yeah,
Yeah.
good point.
Yeah.
So another question from the audience is, experience maybe from SMA again, because there's an approval there. I think this question is coming from, was there any compassionate use for non-trial patients between the trials that were done and those results in FDA approval?
Absolutely, and I have to say I dosed the very first compassionate use patient. So there was a patient who had presented to our center, the day that one of the trials closed, had reached enrollment, and so the patient, we ended up starting on an alternative therapy for a period of time, and ultimately I worked very closely with with the team and was able to get compassionate use. So there is a role for compassionate use. There's actually a lot of regulations around compassionate use as well. So we need to make sure that the therapy is safe and has gone through a certain amount of testing before you can get it ready for compassionate use. But yes, it was certainly something that was considered then, and given the devastating nature of Friedreich's, I would think that it would be considered for for use there as well.
Thank you for that. And I guess maybe that leads into another question that we have from the audience, but maybe from a slightly different perspective. I want to be respectful that, we know we can't talk about any one specific program in terms of what people are thinking about for inclusion, exclusion criteria. We're just not there yet. But there are a series of questions back to some of the earlier topics we talked about around trying to select a cohort and having a homogeneous cohort. And so, is there a sense that, all of the trials might have to be in newly diagnosed individuals or in late stage individuals, or, is it possible? I guess I'd raise the question from two perspectives is, do we think it might be possible that different clinical trials are going to have different types of criteria that might cover different spectrums of the disease? And I guess maybe the other thing I was going to say was, I think it's that compassionate use option or expanded access options allow us, when we have to test a homogeneous cohort, right, to start to expand our experience with a wider cohort. And that can also be helpful. There can be a lot we can learn there.
Mm-hmm.
Yeah, Jen, I think a lot, as you say, it's going to depend. And I think each trial may be different, again, depending on the target tissue and what the goal of the gene therapy might be. For example, if someone has a gene therapy that's specifically targeting the heart and cardiomyopathy, that may lend itself to an entirely different patient population than another gene therapy might. I think it's hard to say right now in general if it'd be only early onset or early more advanced patients. I think it's hard to say. I think we know that the ultimate holy grail, I think Ron Bartek has shared this with me many times. The holy grail will be we can get gene therapy, get newborn screening, and try to treat people as early as possible to prevent any of that neurodegeneration. But that being said, there's still a lot of people who need a therapy who may benefit from gene therapy, and I think that, as is always the case when we're bringing therapies forward, our ultimate goal is to provide it to as many people who may benefit as possible.
Yeah.
And tagging on to something that Meredith had mentioned about, if cells have been lost, we're not going to get them back. And so I would think that some of that decision is also in terms of disease progression. If you're looking for a level of therapeutic effect, you have to take that into account where you're actually dosing patients in a clinical trial where you're able to get that data and be able to see a difference from baseline, essentially.
Mm-hmm.
Yeah.
Go ahead, Meredith.
Well, I was just going to say, bridging on what Matt said, right? I mean, the goal is to be able to get the therapy to, to anybody who is going to benefit, right? So, if somebody, truly isn't going to benefit from treatment, we don't want to take the risk of giving the therapy either, right? So, this is, it's a new therapy and we don't know always what's going to happen. And so you have to make sure that, you do your best you can, right, to make sure that we're not going to make patients worse either. Now that being said, I know there's lots of patients that, if we could, stop progression of heart disease, right? Even if there's been some neurodegeneration, that is really important, right? And so, the goal, will be to make sure to try and, look at the broad patient population to make sure that, we're able to identify who's going to benefit from it and who might not.
Along those lines, Meredith, Ron just typed in a follow-up question, which is that, how early, in some of our traditional drug therapy trials, they've always started with adults, and we've had a lot more adult trials in FA than pediatric trials, and so Ron's asking if there's feasibility to have early pediatric trials and younger children involved in gene therapy trials.
Yeah, I mean, there is, there's additional items that have to be considered, when you're dosing in a pediatric population. So, we haven't touched, and we could have a whole session on informed consent. And I think, this will be something that we'll need to be talking to the community about more as we continue to talk about gene therapy, right? Because in a trial, you're signing up for something that's experimental. And so it's really important that you are well informed going into that trial and that you agree to the risks and the possible understanding of the possible benefits that you might get from the therapy. And so when you are an adult, when you're 18, you're able to sign that consent form yourself, right? You're able to understand the risks that you're taking, and you say, yep, I'm willing to accept these risks, and I understand that this is what I might get from the trial. When you go into pediatrics, the parents sign that consent on behalf of their child. But as in Friedreich ataxia, there's a lot of teenagers and older children that have Friedreich ataxia as well, and it gets more complicated, right? There is what we call an assent form, that we try to make sure that the child is on board as well. But we also have to be extra careful that we really have a therapy that we think is going to directly, benefit, when you treat children. So, I think the goal, ultimately is to be able to as, Matt and others have said, ultimately to get into treating patients as young as we can. But you have to make sure that those extra safeguards are in place, when starting and doing clinical trials in the pediatric population.
I think that brings to a very good point in terms of when we think about continuing education on the topic of gene therapy. We have to continue to think about expanding this knowledge to all levels of understanding, because once a trial is presented to the community for enrollment, we have to be ready that those people that are eligible for that trial really have come to the table with some background and some understanding to be able to fully participate in that consent process.
Yeah, I mean, if you think about it, right, this is one of the only therapies where you're consenting for life, right?
Yeah.
And it's really, really important that you understand what you're consenting to.
Mm-hmm. Very good. We've reached the top of the hour. Jen, do we have any other last questions that we want to ask?
So we do have a few more questions. I think we've covered a lot of them. I think that the part of the sentiment, I think, from the questions is that folks are excited about gene therapies for FA, and for them to be coming to the clinic and just anxious to continue to see the field move forward and be at a place where we can start to be talking even specifically about what FA gene therapy trials are going to look like. And so, I appreciate very much everyone's participation and a lot of the questions that we got from our audience. There are a few I didn't get to answer or get to ask yet, but I'll try and follow up with those after as well.
Mm-hmm.
And anybody can, if they think of additional questions, you can go ahead and submit them to this email, info@curefa.org, and we'll also be happy to follow up with you.
And I just wanted to say, I feel like, we talked about a lot of like really scary things today, but I think what we didn't necessarily talk about is, just how amazing this technology is, right? And the hope that it provides, right? And I have watched gene therapy, being infused and, just thinking like, oh my gosh, this gene therapy is going in and it's getting into the cells and it's replacing this gene that's defective. And, part of the reason that I went from, academic medicine, over to Novartis was really because of the amazing effects that I saw from gene therapy. And so, our goal is to be able to get gene therapy for Friedreich's and other diseases out there. So there's just incredible hope, and what it's done for some of the diseases already has really been incredible. So not to to be such a negative Nelly, we're really excited.
As we said earlier too, it's the FA community can only benefit from all this knowledge that's being accumulated from experience in other diseases. So we're just glad to be part of the process of consideration as a treatment. And we really look forward to continuing to partner with you as those programs possibly get developed.
Same.
So thank you, everybody. Have a good rest of the day or a good evening. And again, please submit any additional questions to this email and we will be happy to get back to you. And thank you very, very much, Meredith and Grace and Matt. It was a real pleasure and honor to have you on this panel.
Thank
you so much.
Good night, everyone.
Bye.
Night.